Okay, hello to all of you. Let's discuss. Basically, this is the last lab, and it is actually a set of three labs. And from this lab, if it would have been a regular semester, we would have written a lab report. This is actually a mini project. So basically, in a regular semester, we would have done. Uh, we would have spent 3 weeks doing this mini project, and you would have written a lab report. But looking at the situation, what the way we are going to do this week, basically I want you to just go over this lab, read the material and watch this video which I'm making right now, because I'm going to tell you some of the important things in this lab. And next week I will provide you with some data. I will provide you with some gel picture, and I would like you to go over that picture, and I will be creating a quiz for you guys on D12, and I would like you to take that quiz on D12, and you can expect similar kind of questions on your lab practical exam. And we'll be finishing labs next week, and the following week you have your lab practical exam number 2, which will be conducted on D12. So let's talk about this lab. So this lab is basically about isolation of proteins from spinach. Now you know that spinach is basically green leaves. So it is very rich in a protein called rubisco. So rubisco is basically an enzyme. So if you know photosynthesis, you know what rubisco is, where it is involved. And the important thing to know, it's about rubisco, it's written here. And the important to know about rubisco is when we will be, so this protein, when you run on the gel, on the polyacrylamide gel, you can expect two polypeptide chains. One is of 55 kilodaltons, another is of 14 kilodaltons. So 55 kilodaltons and 14 kilodaltons. These are the two major polypeptide subunits which we expect to run on the gel. Okay, so let's go over the material for this lab. Basically, this lab is about the isolation of the protein from spinach leaves, and which specific protein we are isolating, that is rubisco. So so in this project is divided into 3 weeks. In the first week, basically we isolate proteins. Okay, so we use the method called ammonium sulfate precipitation method. So this technique or this experiment basically helps us in isolating the proteins from the spinach leaf. So in the spinach, there are different kinds of proteins, but our interest lies in isolating rubisco. Okay, so in the first lab, basically there is isolation of proteins from spinach using ammonium sulfate precipitation. And in the second lab, because we have collected some proteins, in the second lab we purify the protein. We purify the protein and we try to get the rubisco. Okay, and that is done by a technique called ion exchange chromatography. And in the last lab, we do protein gel electrophoresis. So we run the protein gel and we try to see on the gel if we have isolated rubisco or not. So remember that the size of rubisco is 55 kilodaltons. You expect two polypeptide chains on the protein gel. One is 55 kilodaltons, another is 14 kilodaltons. So let's go over the protocol. So okay, let's first of all talk about what you would have done during the first week. The first week you would have isolated proteins. Remember that it will be a mixture of proteins from spinach by using ammonium sulfate precipitation. So for that, what you would have done, so I would have taken 50 grams of the spinach, and then we have two buffers here. One is buffer I, another is buffer A. So buffer I has this potassium phosphate buffer. It contains EDTA, it contains polyvinyl polypyrrolidone. So this is buffer I, and then we have buffer A, which contains tris and EDTA. So what you would have done, so you would have taken these spinach leaves, and you would have taken every group would have deribbed around 50 grams of the spinach leaves, and then you would have collected the spinach leaves from the grass from all the groups, and then you would have blended these leaves in buffer I in the blender, and you would have made a spinach smoothie. and then you would have filtered this spinach smoothie through this mirror cloth, and then you would have collected the filtrate, and you would have saved some filtrate on ice. We will be collecting all the fractions during the experiment because we will be requiring to run all these fractions on the gel during last week. So you will you collect the filtrate, and that filtrate you will be added ammonium sulfate at. So you will be required to calculate the rate you need to add to reach to 37% saturation. This is the amount of written. So after you collect the filtrate, you would have divided the filtrate among different groups, and then every group would have around maybe around 40 ml of the filtrate, and you would have added ammonium sulfate according to this calculation here. The calculation is given here to reach the 37% saturation. So you take that filtrate in a beaker, you take the stirrer, and on the you keep the filtrate on the magnetic stirrer. You put the magnetic bar so that you will be to this filtrate, you'll keep on adding this ammonium sulfate, and you will keep on stirring the filtrate with ammonium sulfate for around 15 minutes. And after that, what you would have done, you would have taken this filtered ammonium sulfate, you would have taken that in the centrifuge tube, and you would have spun this filtered ammonium sulfate for around 15 minutes in the centrifuge at this speed. Okay, so after 15 minutes, when you take this tube out of the centrifuge, you would see a pellet. You would see a pellet, and you would see a supernatant. So the pellet, this pellet is P1 pellet, and the supernatant which you have, that is your S1 supernatant. You can collect some part of the S1, and then you can collect like 1 ml of the supernatant. You could collect the 1 ml of the supernatant in the tube, and you can take this supernatant in the beaker, and then you will, to this supernatant, you are going to add again ammonium sulfate to reach 50% saturation. And again, the formula is given here that you would have used to calculate how much ammonium sulfate to add. So in the beaker you have S1, you will be adding ammonium sulfate, and you will keep on stirring. You will keep on stirring the supernatant. You will keep on stirring the supernatant 1 with ammonium sulfate for 15 minutes. And after 15 minutes, you will collect this supernatant 1 with the ammonium sulfate in the centrifuge tube, and you would spin that down in the centrifuge at this much speed for 15 minutes, and then after 15 minutes you will have a pellet. Now this pellet is called P2, and then you have S2. The supernatant is S2. So basically, reaching up to this point, so this is basically experimentally what you would have done during the first week. So at this point you have P1, you have P2, you have S1, you have S2, also you have F, which is a filtrate. Now with P1 and P2, these are the pellets. You would dissolve these pellets, both P1 and P2, into water. So the volumes are given, how much to dissolve, and then you would have taken dialysis bags, and in the dialysis bags you would have loaded these diluted pellets, and you would have dialyzed these pellet number 2 against this buffer A for around 3 to 4 days. Okay, now I'm going to explain you conceptually, experimentally, scientifically what we have done in this experiment. So we are using ammonium sulfate precipitation. Now ammonium sulfate precipitation, what is the purpose of using ammonium sulfate precipitation? And there is also one question here. So pay attention to all the discussion points, because these are the kind of questions which will be asked on your quiz. So ammonium sulfate, why we are adding? So in the spinach leaves we have these proteins, and we have made this kind of so these proteins, they are actually forming bonds with water. Okay, so by adding ammonium sulfate, what we are trying to do, we are trying to add the salt so that the salt can make the bonds with water and does not make the bonds with proteins, so that the proteins, they actually precipitate out. Okay, so the proteins no longer make bond with water, and the water actually makes a bond with salt, and the proteins, they precipitate out. That's what we are doing. and we expect the proteins in the P1 and P2, because because we spin we so we take the supernatant and we add the salt, and the proteins which will be precipitated, they will be in the pellets. So these pellets, we dissolve them in water. We dissolve them in the water, and we take the dissolved pellets in the dialysis bags, and we do the dialysis for 3 to 4 days. So what's the purpose of doing dialysis? The purpose of doing dialysis is because in the P1 and P2 we expect a lot of salt, okay, and by dialysis we are trying to get rid of the salts. Now these dialysis bags, the dialysis bags are of different pore size. It means these bags, they have pores in them, and through these pores the some salts can come out. So the purpose of doing dialysis is to get rid of these salts. So okay, so what we would have done in the second week, so by the first week you would have the lab by putting P1 and P2 in the dialysis bags, and you would have had these, you would have you would have filtrate, you would have S1, you would have S2, and these would have been all stored in the fridge. So next week when you would have come, you would have done the purification of the protein. So in the P1 and P2 you have mixture of proteins. Now we want to purify them. So how to purify them? So we would have passed the P1 and P2 over an ion exchange column. Okay, now what's the ion exchange column? So ion exchange column basically works on the principle that these columns, they are made of saffrose. They are made of these some kind of beads, and these beads are made of saffrose, and so they have, so in the column there is a resin. Okay, so there's a column, in the column there's a resin, and the resin has beads which are made of cellulose, and these beads, they are covalently bound to positively charged side groups, so that when we pass the mixture of these proteins, over this column, because this column is positively charged, all the negatively charged proteins will be binding to the column. All the negatively, and ribisco is a negatively charged protein. Okay, so basically for this for the purification we use ion exchange chromatography, and we use the saffrose fast columns to perform the ion exchange chromatography. And these columns, they have in the column they have a resin, and this resin is made of tiny beads of cellulose which are bound with some positively charged groups, and we pass the protein mixture over this column, and the negatively charged proteins, they go and bind, the negatively charged proteins, they go and bind to the positively charged column. And this is the way how the proteins, they negatively charged proteins, they get bound to the column, and all the other proteins, they're going to pass through. And then after that we use the salt buffers to elute these proteins from the column. So let's go over the protocol how we would have done. So you would have taken these columns, so you would have so every column has these 4 parts. It has a plastic column, filter, stopcock, and the lid. So basically you would have got this column, and first of all you would have equilibrated this column with the buffer A. So you would have equilibrated the column with buffer A, and for that basically you pass around 30 ml of the buffer A. It goes through the column. This is called equilibration. And on the other hand, you would have taken your P1 and P2, you would have spun them down so that anything which is some solid precipitator, or that they should all get settled down. And you would have taken the supernatants, P1 and P2. And for P1 you would have been required to dilute this because it has a lot of proteins, and P2 we can take undiluted, and you would have loaded these P1 and P2 on the column. So you would have passed these dissolved pellets in water over these column. Okay, so you will basically what are you doing here is letting these dissolved pellets to go through this column, and all the negatively charged proteins which are there in the pellets, they're going to go and stick to the column because the column is positively charged, ribisco is negatively charged. And after that you would have washed the column with buffer A, and then after that we have three different kinds of buffers. One is a low salt buffer, then we have medium salt buffer and a high salt buffer. Now what these buffers are? These buffers are basically buffer A. These are basically buffer A with some salt in them. So the low salt buffer is basically buffer A with 50 millimolar sodium chloride. Then we have medium salt buffer, which is buffer A with 200 millimolar sodium chloride. And then we have high salt buffer, which is buffer A with 500 millimolar salt. So after you have washed the buffer, you would be passing first of all the low salt buffer, then the medium salt buffer and the high salt buffer. And once you're passing, so you pass these buffers one by one and you collect the fractions. You collect the fractions. So in the fractions, you get the eluted protein. So why you are able to elute with these salt buffers? Because in these salt buffers, we have three different types: low salt, medium salt and the high salt. So in these buffers, we have sodium chloride, and sodium chloride has two kinds of ions. Sodium is cation plus and chloride is negative, it's anion. So when you pass these low salt and medium salt and high salt buffers over the column, the chloride ions, which are also negatively charged, they are going to go and take the place of protein, and the protein is going to come out of the column. Okay. So you will low salt, medium salt and high salt, they are just the same buffer with high concentration of sodium chloride. And sodium chloride is a salt which has two kinds of ions, sodium plus and chloride negative, chloride anion, which is negatively charged. So when you pass these buffers over the column, the chloride ions, they're going to go and stick to the column. They're going to take the place of the negatively charged proteins, and the proteins they're going to elute from the column. And those fractions you collect, and then you check the OD by the spectrophotometer, and we check the OD in the ultraviolet radiation range, in the UV range, which is OD 280, because in these proteins they have some of the aromatic amino acids like tyrosine, tryptophan, phenylalanine. These are all aromatic amino acids. So we check the OD in the UV range using the spectrophotometer, and on the basis of the OD, we can actually select some of the fractions and we can discard some of the fractions. So usually if the OD is more than 0.1, we keep the fraction. If the OD is less than 0.1, we usually discard the fraction. And the last week, so you would have collected all the fractions, you would have kept them in the fridge, and next week what you would have done, you would have run the protein electrophoresis gel. So these gels are usually made, so these are vertical gels. You must have run the DNA gels. Those are the horizontal gels, and you know that in the DNA gels there are different wells in which we load the samples. So there are different wells. In which we load the samples. So similarly, in the protein gels also there are wells, and this gel is vertical and the DNA gels are horizontal. So in these gels, this is made of a material called polyacrylamide. Also in this gel, STS is added. Now what is STS? It is a detergent. That is why this is called STS page. This is called so STS is a detergent, sodium dodecyl sulfate. So basically sodium dodecyl sulfate with a detergent has a negative charge. So when we pass the proteins through this gel, all the proteins, they get denatured. Okay, all the proteins get denatured because STS is a detergent. So all the proteins get denatured. You know that proteins, they have a three-dimensional structure. So denaturing means they all lose all the folding and the protein is converted into polypeptide chain. They all become straight chains. So that is what we are doing. So STS denatures the proteins, as well as STS is negatively charged, as well as these proteins, these polypeptide chains, they also get negative charge on them. So that is why when these proteins, these polypeptide chains, these denatured proteins, when they run on this gel, they run on the basis of their molecular weight and not on the basis of charge, because the charge on these polypeptide chains become negative because of the presence of STS. So STS actually masks the charge on the proteins, and it gives all these polypeptide chains negative charge. Okay, so basically, how do we run the gel? So the gel is already pre-made. So we take all the samples. Okay, so we at reaching at this point, we have filtrate, we have S1, we have S2, we have P1, we have P2, we have like this P1 and P2. We have actually we also save before we pass the P1 and P2 over the column. We save some, and also we have collected the different fractions of P1 and P2 after passing through these columns. So we have collected those also. So we take these samples and we add a loading buffer. Okay, what is a loading buffer? So loading buffer has a dye called bromophenol blue, glycerol, dithiotretol, and STS. So bromophenol blue is a dye which is added to these protein sample. So basically it tracks the proteins in the gel. And dithiotretol is a reducing agent. It actually breaks all the disulfide linkages in the proteins, and it helps in proteins to become all the straight chains, because dithiotretol is a reducing agent and it breaks all the disulfide linkages in the protein. So it's a very important point you must know, that dithiotretol is a reducing agent. It breaks the disulfide linkages, because all these disulfide linkages, they help in the it helps the proteins in attaining that it helps the proteins in attaining their native conformation, or it helps the proteins in attaining their three-dimensional structure. So that's the function of dithiotretol, which is a reducing agent. It breaks the disulfide linkages. And STS is a detergent. STS is there in the gel also. It is also in the this buffer, sample buffer also. So basically it is STS, it basically denatures the proteins. STS denatures the proteins. So basically we mix these samples and we heat over a heating block for 5 to 7 minutes. Heating is again trying to help the in the denaturation of the proteins. It again helps in the denaturation of the proteins. So this is the way we prepare the samples, and then we load these samples on the gel, and then this gel is connected with the power supply, and then we run it for around 45 minutes to 50 minutes. And once the gel is run, we take the gel out and we stain it with Comessi blue. So Comessi blue is a kind of a stain. So we keep the gel with this stain for basically we can keep the gel with the Comessi blue for 5 to 7 minutes, and then we can actually destain with water. There are different ways of staining the gel. Sometimes we can even take a Comessi blue and we can just microwave for 30 seconds. The gel stains very well, and then you can destain it. But in this case, if we microwave, I think it takes a very, very long time. So even we had a stain in which, and then we can destain with water. It's quite quicker. So basically overall the thing is, after you have run the gel, you have to stain with the dye, with the Comessi blue dye, and then we destain it. And after you destain the gel, you can see the different bands in the different fractions, and that's what I'm going to give you the data for. That's the gel pictures I'm going to give you for the next week. So this week I want you to read this lab, watch this video, and their discussion points. Make sure you understand all the discussion points. And also when we run we also run a protein marker with other samples. Now what is the protein marker? Basically we buy the marker from the companies, and protein marker is a kind of a reference in the gel. So in the protein marker, we know that when we run it on the gel, we see the 7 bands and what is the exact size of each band, so that we can compare the size of the other unknown bands in the gel. So protein marker is kind of a reference. So please go over all the discussion points this week, and I will be providing you with some gel pictures for the next week, and you will be also answering a quiz on D12. If you have any question, please send me an email.